Good day, I am Princess Nurfida Nur Mangelen, and together with my group mates, Francesca Benedian and Lauren Orong, we will be presenting to you a brief report regarding 2D SDS page. The principles behind the 2D SDS page. The 2D classic method was first described by O. Farrell in 1975. It utilized acrylamide tube gels polymerized with commercial carrier amphylines for IEF. Voltage application creates a pH gradient, and the overnight IEF step causes amphylines and proteins to move to a steady state location. The resolution of hundreds of proteins was made possible by this. Meanwhile, 2D SDS page is called such as it employs two dimensions or steps according to two different properties for separating proteins. Isoelectric focusing, which separates proteins based on their isoelectric points, is the first dimension. Meanwhile, the SDS polyacrylamide gel electrophoresis or sodium dodecyl sulfate polyacrylamide gel electrophoresis, which separates proteins based on their molecular weight, is the second dimension. The principle of isoelectric focusing. In the presence of a pH gradient and electric field, proteins with different isoelectric points will migrate until the net charge of a protein is zero or neutral, at which time the migration will stop. Meanwhile, the principle for SDS page. Proteins have varying charges depending on their amino acid composition, so samples need to be treated with sodium dodecyl sulfate to make them all negatively charged. If this is skipped, their migration behavior will vary as some will be moving upward instead of downward or toward the end which has a positive charge. Moreover, proteins have different shapes that can be a factor in the way they will migrate the gel matrix that has pores. Thus, SDS also aids in making their shape uniform and cylindrical. In this dimension, the proteins with a higher molecular weight will be found at the top as they will have a shorter migration distance. Moreover, proteins with a higher molecular weight will travel slower and will not easily traverse the pores of the matrix, unlike those with low molecular weight.
Now let's proceed to the procedure of SDS page. First is the preparation of gel. To prepare the gel, you must set up two different types of glass plates, a fully square one and one with a slight depression. And we will also prepare combs that will create the wells where the sample will be placed or injected. We will also need silicone tubing to prevent our liquid monomer solution from leaking when we press the two glasses together. In addition, binder clips will be used to secure the two glasses together. Pour the monomeric acrylamide solution after preparing the glass plates. Then, distribute a thin layer of water or ethanol so it evens out the polymerization. So when the gel polymerizes, there is a horizontal straight layer. If neither water nor ethanol is added, the gel polymerizes and becomes non-adhesive. We will not place the comb here because we must first prepare two layers, which is the running and the stacking gel. Once the running gel has polymerized, the ethanol or water layer must be removed and the stacking gel must be poured. You have two distinct acrylamide concentrations. After dispensing the stacking gel, incorporate the comb to form the injection wells. After placing the comb, one must again wait for the gel to polymerize. This process takes approximately 45 minutes to complete. For the first dimension of our procedure, we will execute the isoelectric focusing. And in here, first the protein is loaded at the top of a column where pH is very high. And most of them are negatively charged at this pH, so the protons are stripped from residue side chains. Then the proteins move in the electric field toward the distant cathode and away from the nearby anode. And as the proteins move through the pH gradient, they gain positive charge and reach neutrality. So the proteins stop exactly when pH is equal to Pi and the stained proteins are very visible. Highly stable ampholytes are molecules with specific pKa to give a specific and unchanging pH gradient. So the separation is achieved by applying a potential difference across a gel that contains a pH gradient and the procedure, the isoelectric focusing, requires solid support such as polyacrylamide gel, which will be executed in the second dimension of the procedure. Now let's proceed to the procedure of SDS page. First is the preparation of gel. To prepare the gel, you must set up two different types of glass plates, a fully square one and one with a slight depression. And we will also prepare combs that will create the wells where the sample will be placed or injected. We will also need silicone tubing to prevent our liquid monomer solution from leaking when we press the two glasses together. In addition, binder clips will be used to secure the two glasses together. Pour the monomeric acrylamide solution after preparing the glass plates. Then, distribute a thin layer of water or ethanol so it evens out the polymerization. So when the gel polymerizes, there is a horizontal straight layer. If neither water nor ethanol is added, the gel polymerizes and becomes non-adhesive. We will not place the comb here because we must first prepare two layers, which is the running and the stacking gel. Once the running gel has polymerized, the ethanol or water layer must be removed and the stacking gel must be poured. You have two distinct acrylamide concentrations. After dispensing the stacking gel, incorporate the comb to form the injection wells. After placing the comb, one must again wait for the gel to polymerize. This process takes approximately 45 minutes to complete. So while the gel polymerizes, you should begin to prepare samples. First, we place protein samples in a microfluidic tube and add sample buffer and mix by flicking. The ratio should be 1:2, which means if you have 5 microliters of sample, you should add 10 microliters of buffer. Sample buffer contains SDS, bromophenol blue, which is a tracking dye so that when electrophoresis is performed, you will know that the protein samples have reached the bottom, and 2-mercaptoethanol, which is essential because when preparing samples for SDS page, we must completely denature them down to their primary structure. And 2-mercaptoethanol cuts disulfide bridges between the two cysteine residues and reduce them to thiol. The samples are then heated at 100 degrees Celsius for 3 minutes to ensure complete protein denaturation. Then, centrifuge the sample for 1 minute to remove any debris that could obstruct the pores of the gel matrix. Lastly is the electrophoresis. First, we remove the gel from the casting stand and place it in a glass plate to appropriately position the gels in the electrophoresis apparatus. The two glass are different. When put together, the glass plate with a depression should face inward so that the tank buffer can penetrate when poured into the tank. Afterwards, pour running buffer into the upper and lower chambers of the electrophoresis apparatus and remove air bubbles and small pieces of gel from the wells and under the gel using a syringe or tips of the pipette. Then, pipette a specific amount of the denatured sample that was prepared earlier. And then we turn on our apparatus power supply and run the gel until the dye in the sample buffer reaches the bottom of the gel. Remove the gel assembly from the electrophoresis apparatus when completed, and using a spatula, remove the gel from the glass plates and stain them to visualize the banding patterns. The gels are stained by submerging them in a solution containing Comazine blue, acetic acid, and methanol in the proportion specified. And once the gel has been tainted for 24 hours, it must be destained with acetic acid and alcohol for another 24 hours. And after this procedure, we are now prepared to conduct subsequent analysis.
Sample data analysis. In order to study mucopolysaccharidosis type 1 or MPS1, which results in an accumulation of glycosaminoglycans that have not been undegraded due to deficiency in alpha L iduronidase, Wu et al. In their paper entitled Proteomic Analysis of polysaccharidosis 1 mouse brain with two-dimensional polyacrylamide gel electrophoresis employed 2D SDS page. Using 2D SDS page, the brains of MPS1 and control mice were examined in order to find possible biomarkers. 2,055 protein sites in total were compared. Of them, 25 locations with 50 distinct proteins and a fold change of more than 3.5-fold and a p-value of less than 0.05 were identified using nano-LC MS. The MS spectrum of protein 1351, sirtuin SIR2L2, is displayed in the figure presented. In Figure A is the MPS1 brain sample's 2D silver stained difference pattern. Spots that are increased in MPS1 compared to the control are highlighted in blue, while those that increased in the control are highlighted in red. Discussing the results, it was found that the MPS1 and wild-type mouse brains expressed 50 proteins differently. These proteins participate in a variety of biological activities. Firstly, cytoskeletal proteins like actin, dynamine, actinin, tubulin, and dynein make up a significant subset of dysregulated proteins in the MPS1 mouse brain. The proteins that are involved in metabolism, like GLS, are another class of dysregulated proteins in the MPS1 mouse brain. Additionally, the expression of numerous proteins involved in neurotransmission, like DPYSL2, and for protein folding, like HSPA2, are downregulated. Meanwhile, protein expression of those for intracellular ionic homeostasis, oxidative stress response, RNA splicing, signal transduction, hydrolase activity, and extracellular matrix are dysregulated. These findings for the first time emphasize the significance of changes in intracellular ionic balance, metabolic pathways, and the cytoskeleton in the neuropathology of MPS1 illness. The proteins discovered in his work could serve as MPS1 diagnostic and potential biomarkers. Sample application. Alpha-synuclein in neurodegeneration was the topic of Wang et al.'s investigation in their paper entitled Protein Arginylation Targets Alpha-synuclein, Facilitates Normal Brain Health, and Prevents Neurodegeneration. It was discovered that alpha-synuclein is an efficient substrate for arginyl transferase and is arginylated in vivo. Also, if there is a lack of arginylation, it could lead to alpha-synuclein aggregation that will impose pathological consequences. The researchers utilized 2D SDS page of a radio-labeled substrate to showcase the arginylated alpha-synuclein. Meanwhile, the native mouse brain's arginylated alpha-synuclein amino acid residues were identified using mass spectrometry. Moreover, to ascertain if milk proteins were impacted by breast cancer, Aslebag et al. In their paper entitled Comparative Two-Dimensional Polyacrylamide Gel Electrophoresis of Human Milk to Identify Dysregulated Proteins in Breast Cancer, used 2D SDA's page with mass spectrometry. Milk from a woman who had been diagnosed with breast cancer was compared to normal milk from the other breast using a silver stained 2D SDS page to look for abnormalities. In comparison to controls, 5 proteins were found to be elevated and 5 to be downregulated in breast cancer. These possible indicators could offer a technique for predicting the aggressiveness of breast cancer. Other applications of 2D SDS page include protein species and isoform distribution analysis, post-translational modification studies, and downstream analysis such as Western blotting.